Hi there fellow primates, I hope this video finds you well on this windy Friday afternoon. Storm Eunice has arrived here in London, and so far we have lost 2 panels of our back garden fence, so fingers crossed we're not going to get more damage. If you're watching this and you live in the UK, especially in the south, I hope you're all safe. Now, let's start with a little riddle. What do the following items have in common? The mouthparts of bumblebees, the feces of mice, the feet of moths, the heads of babies, the wings of butterflies, the anal glands of dogs, cooked horse meat. Any idea? Well, if you want to know the answer, stay tuned. We're going to explore that in today's Journal Club. And the paper I selected today, I picked up in the Twitter feed of Robert Trivers, the famous evolutionary biologist. It's his birthday tomorrow. I think he's turning 79 years old and he's still doing active research. So that's fantastic. If you're watching this, happy birthday, Dr. Trivers. And if you're not aware of Robert Trivers' work, he is really famous. He established his reputation in the 1970s with hugely influential work on the evolution of reciprocal altruism, parent-offspring conflict, more recently the study of symmetry, body symmetry, and how it relates to athletic ability in humans, the evolutionary logic of what we call honor killings, and he's been working on establishing the foundations of a theory of deceit and self-deception. And on this topic he has written a book from 2011, a wonderful book called Deceit and Self-Deception, Fooling Yourself, the Better to Fool Others. And that in a nutshell is the theory that he elaborates in this book. That is that self-deception is not a flaw, it's actually useful adaptively, because if you are better able to deceive yourself, then you can very easily... Whoa! I just saw the window opposite open very forcefully in the wind, and now curtains are coming out, and all kinds of debris seem to be being sucked out of that bedroom. So that's a bit unfortunate. I don't think these neighbors are home, so I hope that's not going to cause more damage. I'm not going to try to stop and re-record this. This is just action happening right now. A bit exciting. The tree in front of my window is now full of pieces of plastic and what look like ribbons that may have been part of a present. Well, yeah, the storm is continuing. So back to Robert Trivers, this book about deception and deceit and self-deception. So he argues that self-deception evolved as a trait because that allowed you to better deceive others as well. So this is a really interesting book that I recommend you all reading. And he has led a very interesting life as well. There's another book that he published, I think 2015 or 2015, titled Wild Life. And that is subtitled The Adventures of an Evolutionary Biologist. And adventures Robert Trivers certainly has had in his career and his personal life. If I just read one sentence from the back cover to give you some idea. Not only is he a famous scientist, one of the most original and creative thinkers in biology I think there is, his life has had some high points that most of our lives don't have. So just to read that sentence, unlike other renowned scientists, Trivers has spent time behind bars, drove a getaway car for Huey Newton, who was the co-founder of the Black Panthers, and founded an armed group in Jamaica to protect gay men from mob violence. So these are just a very few of the ingredients of one of the coolest memoirs, autobiographies that I've ever read, certainly by a scientist. It reads a little bit like if Hunter S. Thompson had been a scientist and he would write his memoirs, you would get something akin to this. Now, the paper that I picked up on his Twitter feed is a very interesting paper. But before I get there, I just wanted to... I was a bit distracted now by all the debris flying around again, so it's going to be perhaps a bit of a bumpy journal club. I met Bob Trivers once. I remember it was in November 2017, and I went down there to Madrid to visit my friends Christoph and Maite and to give a talk at the Autonomous University in Madrid. And my wife Sarah and I were put up in university accommodation, and at some point in the morning we were having breakfast, and I see Robert Trivers sitting there at the table. So fanboy that I am, I walked up to him and said, "Oh, Mr. Trivers, is that you? Are you Robert Trivers?" He said, "Yes, yes." I said, "Oh, well, I'm a fan of your work, loved your books, fantastic, so nice to meet you." Spoke with him maybe 10 seconds, went back to my table because he was in conversation with someone else having breakfast. When his partner left, he came and actually sat with us. So we spent maybe 10 minutes or so chatting about our lives and work and why he was in Spain, which was a really interesting experience. And then we bumped into him later that day as well. And we were there with Christoph and his little daughter Clara, who you can see here in this photo. This is a photo taken when we were having food with Christoph and his wife Maite and their little daughter. And as you can see, Clara is the cutest little thing, was the cutest little thing at the time. And when we were there at the hotel, we bumped into Bob Trivers again. And he went up and, of course, said, "Oh, that's such a cute little girl." And as he was standing close to her and admiring her cuteness, this little fist came out and she punched him right in the face, which was a bit surprising. But he didn't retaliate. He kept smiling. He was calm, despite the fact that when you read interviews with Trivers himself, he admits to having some anger management issues. So, but nothing happened. And maybe because, it was a little kid. If an adult had hit him, then physical violence would probably have ensued. But there is something special about little kids, especially about babies. Allow me to illustrate. You can see in these pictures here, a younger me, I think I was 28 years old here, and I had little Trevor on my lap, the son of my friends Pascal and Corina. And when Trevor was born, at the time I lived in Amsterdam, and I was doing my PhD at the Zoological Museum there, I got a call. "Yes, the baby has arrived. Could you please come down and bring us a nice little jar with alcohol?" Because Corina wanted to preserve part of the umbilical cord of the baby. So it's always handy to have a friend in the museum. So I cycled down there and handed off the jar with alcohol. And I remember being offered Trevor, so I held him while he was still extremely young. And I remember cuddling him and going, "Ah, he smells so wonderful. There's something special about baby smell." And I remember Corina looking at me. It's like, "Yes, Ronald, he has not been washed yet." And it's like, "Oh, of course not. He just came out of you." And up to today or this week, this was sort of a funny, relatively embarrassing little story, until I bumped into the paper that we're going to discuss now. There's perhaps something more to be said about baby smell. So the paper I want to discuss here was published in Science Advances in late November of last year by Eva Mischur and an international team of researchers, most of them colleagues of her at the, I have to look, Weizmann Institute of Science in Rehovot in Israel. And the title of the paper is "Sniffing the Human Body Volatile Hexadecanal Blocks Aggression in Men but Triggers Aggression in Women." And this is a paper completely outside of my field of expertise, but really interesting because it is such a fascinating thing to realize that there may be chemicals in the world that you can't easily detect, maybe not at all consciously, and yet they can have an impact on your behavior. And that's the topic of this particular paper. And they set the story up by noting that chemical signals play an important role in modulating social behavior in animals. So, for instance, they cite the example of a peptide that is found in the tears of mice. And when you use that peptide and you put it on baby mice, then males, the aggression towards the baby mice is blocked. Naked mole rats, they have an eye-associated gland called the Hardinian gland. And they use the secretions of that gland to rub them on themselves, and that suppresses aggression towards them by more dominant males in the colony. And it's even been shown that for human males, when human males sniff human tears, their testosterone levels come down. And it's not known if that translates also into a decrease in aggression towards other humans, but it is in that sort of realm of ideas that Eva Mischur and her colleagues came up with this question, and I can read it from the paper. Could human aggression mechanisms be tied to chemosignaling mechanisms as they are in all other terrestrial mammals? Because they note that, using mice again as an example, mice have a receptor for this chemical, hexadecanal, and this is the chemical formula here. Hexadecanal is a long chain aldehyde, and it is a compound that in larger concentrations has a sort of sweet odor, but in low concentrations you cannot really perceive it. And that plays a role in the social behavior in mice. It seems to modulate social interactions. It seems to work as a social buffer compound. And the receptor for this particular chemical, a homologue of that, is present in our genome as well. And in fact, previous research has shown that a homologue of this hexadecanal receptor is present throughout mammals. So it is a conserved evolutionary feature, and hence the question arises, well, could it play a role in receiving, in transducing chemical signals, and let me rephrase this, could hexadecanol play a role in humans via this receptor and modulate human behavior after they have been exposed to the chemical? So these authors did a really sophisticated study. So it's a statistically intricate study in which they used a number of test subjects, both women and men, and then they did two particular kinds of experiments in which they created a scenario in which aggression was provoked, and then the test subjects were given an opportunity to vent their aggression, retaliate, take revenge. So in this particular schematic, you can see one of these experiments where control subjects for both women and men were given basically a control compound to sniff, and the test subjects were given hexadecanol to sniff, and then they were engaged in a game in which they could make money. But then they were playing against the person who cheated them, and that of course caused aggravation and provocation of aggression. And then they were given a chance to basically punish the player that they played against by using loud sounds, so very annoying sounds that they could choose different intensities of. So that was a way in which the test subjects could be tested for the build-up and release of aggression, and then see if sniffing hexadecanol had an impact on this type of behavior. And very, very strikingly, it does, but in opposite ways in males and females. As you can see in this particular graph here, you can see the experiments on men in the top and women in the bottom across 16 different trials of this game. And you can see, if you look at the top left graph, the control for men, that the distribution of noise blasts given with a color coding of intensity, with the dark colors being the most intense across the 16 trials. And then when they were exposed to hexadecanol, you can see a marked decrease in the intensity of the noise blast, of the painful noise blast, the punishing noise blast given in the test subjects. And this was a strong effect that you could see here. So, hexadecanol seemed to make the men less willing to give punishing noise blasts to their game opponents. And then in the two lower panels, you can see that in women it was completely the opposite, where the control group has the lower left panel of a distribution of noise blast intensities. And then when they're exposed to hexadecanol, they start to give more and more intense noise blasts as punishment. So it seems that women, after exposure to hexadecanol, become more aggressive, more willing to punish somebody they think wronged them. This particular graph shows you the same idea and the same data, the same results in a different way, where for women on the left and men on the right, the graphs indicate the difference in noise blast intensity delivered to their game partners in the control groups and after sniffing hexadecanol. So if you look at the graph on the left for women, you can see that control women give less intense noise blasts, and then when they're given hexadecanol to sniff, then they become willing to give more punishing noise blasts to their game partners. And in the right graph, you see exactly the opposite for males. So this is a very interesting discovery that women and men respond differently in terms of their willingness to be aggressive to others based upon whether they're exposed to hexadecanol. It makes men less aggressive, and it makes women more aggressive. Now, they went further than this. They wanted to know, is there a neurological correlate? Is there a neurological substrate in the brain that may be associated with these different types of behavior? And indeed there is. So they designed different tests to provoke aggression in the test subjects and then give them an opportunity to release the aggression. And in this case, they were not pushing buttons that gave blasting sounds. They gave them a rubber ball they could squeeze, and they could see that, the harder somebody squeezes the ball as a measure of increased aggression. And this different setup allowed them to do functional magnetic resonance imaging, fMRI. So they could look, while they did these tests, at brain activity in different parts of the brain to see if they could trace the response to aggression propagation and the release of aggression in the brain. And a striking thing emerged. In both women and men, the exposure to hexadecanal, sniffing hexadecanal, activated an area in the left side of the brain called the left angular gyrus. I don't know what it does, but it's apparently a brain region that has been shown to have a role in the perception of social cues in humans. So that's both true for men and women, as you can see in this particular picture here. You see the little orange dot in 3 of these 4 panels, and that is located in the posterior left side of the brain, of the cortex. But then they ask, well, if you look at further activation of brain regions in men and women, are there differences? Because men, of course, and women respond differently to hexadecanal. And they try to map the connectivity between the brain center that becomes activated due to exposure to hexadecanal and associated brain centers that have a role in the execution of aggressive behavior, such as the amygdala and the orbitofrontal cortex, and brain regions that have to do with social appraisal, like the temporal pole. And as you can see in this particular graph, you can see that there's difference between the men and women again. So in different colors, the control and the test, the control experiments and the tests of when men and women are indicated. And these 5 abbreviations are different regions of the brain. OFC here is the orbital frontal complex, for instance. And if you look at that one, you can see that the control for women falls on the right-hand side of the vertical line, and the response to hexadecanal on the left-hand side. And that indicates that after hexadecanal is perceived, the link between the left angular gyrus that is activated by exposure to hexadecanal and the orbital frontal complex is decreased. And the same is true if you look at the left amygdala, which is the graph beneath it, or right at the bottom of this graph, the right temporal pole, where exposure to hexadecanal decreases the connectivity of these regions to the left angular gyrus. And for men, it's completely the opposite, where the exposure to hexadecanal actually increases connectivity between these different brain regions to do with execution of aggressive behavior and social appraisal, and the left angular gyrus that is activated by exposure to hexadecanal. So a lot of information all pointing in the same direction. And I will read out what they conclude from this. And they conclude that, hi, I'm back. I just popped across the street to tell the neighbor that his bedroom window had blown open. Stuff was being sucked out of the room, so he has closed that now. And it was a bit distracting, so with all the stuff flying by, to stay focused on the journal club. So, just to summarize then, Eva Mishor and her colleagues showed convincingly that exposure of humans to hexadecanol has a completely different effect in men and women. It seems to increase aggression in women and decrease aggression in men. And there are neurological correlates there as well, where the region that gets activated by exposure to hexadecanol, the left angular gyrus, increases connectivity to other brain regions that have to do with social appraisal and the execution of aggressive behavior in males, and decreases the connection to these regions in women. And the author speculates that that correlation may be a causal one, in which these brain regions have to do with modulating aggressive behavior in women and men. Of course, more experiments need to be done, but they would have to be done on human subjects, which is a tricky issue. Another caveat is that they didn't know if the amount of hexadecanol they exposed the test subjects to are physiological, whether the amount of hexadecanol that comes off the skin and the breath and the feces of humans, which are the three main sources of hexadecanol, are in the same range. But what is the biological relevance of all this? And they have a speculation, and this comes back to baby smell, what we started today's video with. They discovered as part of this study that hexadecanol is one of the most abundant baby head volatiles. And in all cultures, parents are encouraged to sniff their babies. In women, this activates reward systems in the brain. And so they surmise that there is an ecological evolutionary rationale going on here. That is that when males smell babies, their aggression gets reduced, and that reduces the chances that they harm the baby. And for women, the aggression gets increased by exposure to hexadecanol, making them more willing to fight to defend their offspring. So it seems to make ecological sense from the perspective of the baby to try to appease males, especially males that are not your father, and that and to encourage your mother to defend you against any harm. So that's the evolutionary biological rationale, the hypothesis they propose. So in their own words, they say, in summary, babies emit hexadecanol from their head. And this is expected to trigger aggression in women but block aggression in men, and both of these impacts are expected to increase baby survival. So that's really cool. So they also discussed, now, does that mean that hexadecanol is an actual human pheromone? And they argue that, well, imagine that we did this entire study and test subjects were not men and women but mice, then no reviewer would say that, no, this is not a pheromone. So that they conclude, yes, hexadecanol is a human pheromone. A pheromone that is used by other animals in other ways, which brings us to the conclusion of today's video, and going back to the riddle, what do these things have in common? The mouth parts of bumblebees, the feces of mice, the heads of babies, the wings of butterflies, the feet of moths, cooked horse meat, the anal glands of dogs, well, you can guess by now, they all emit hexadecanol. But in these different cases, the exposure to hexadecanol can have completely different effects. For instance, if you cook horse meat, and in the process hexadecanol is released, and if you mature horse meat, and that apparently is one of the reasons why horse meat can be considered to have a sweet taste and a sweet smell. Same, people who study the impact of cooking methods on the tastes of lamb meat and pork products also have a keen interest in chemicals like hexadecanol. In the world of biology, well, hexadecanol is emitted, for instance, by dead insects that have been killed by entomopathogenic nematodes. These are nematode worms that kill insects and then live on the cadavers. And that hexadecanol that is derived from these killed insects, if you expose ants to it, they don't like it. They don't want to eat these corpses. Bumblebees and bee wolves, which is a species of wasp, they have mandibular glands on their mouth parts that exude and emit hexadecanol, and it is used as a territorial marker. What else? Parasitoid wasps use it as a sex pheromone, as do moths, and as do butterflies. And if you look at this particular beautiful butterfly with the cool name, the African squinting bush brown, this butterfly produces hexadecanol as a pheromone. It is very present, I think restricted to the hind wings of males, but in inbred males, the concentration of this pheromone goes down. And this is a way in which women pick the boys, because in the inbred males with very little hexadecanol, they don't find them attractive for mating. So it's sort of a quality assessment pheromone for female butterflies. So there we come to the end of today's video. This one simple organic molecule present everywhere in the animal kingdom and fulfills completely different roles. So that just goes to show how deep the rabbit hole of biology goes. You type in as simple a word as hexadecanal in Google and the Web of Science, and you get sucked into this whole world that I had no idea existed. And that's just the beauty of biology, and that's why it's a privilege to be a researcher. I've talked long enough. I hope you find this interesting. If so, like it under the video, subscribe to the channel, sign up for alerts. I wish you all a good weekend. In the last 10 minutes or so, the wind seemed to have died down here, so fingers crossed that it will stay like this. Stay safe and see you next week. Bye bye.